How's everyone doing today, though?
I'm doing good. How about yourself?
I'm doing well, thank you. What have you guys thought about the class so far? Sorry, I'm just submitting more people now.
I have a quick question actually.
Hmm.
So like I was going through a lot of the PowerPoints and the reading and stuff, and I'm trying to kind of like actively recall off the top of my head like the information as I go and kind of test myself. But I guess I get
Okay.
concerned with like the pace of the class. When it comes to like exams and stuff, is it gonna be like multiple choice, or you're gonna have to like know everything like instantly, active recall kind of thing, or
So, from my understanding, so this is the first time I'm actually TAing for this course, so I haven't seen it in its entirety. Um, yeah, since it is such a fast-paced course, you are required to spend a lot of time to kind of understand all the concepts. And during the exams, I know you'll be using like this honor log system, which I've personally never used, but some of you may have. Um, And I think, since it's pretty stringent on what you can and can't do while you're taking the exam, like from my understanding, your hands have to be like in like the picture frame almost, and like you can't look like you're looking at previous notes. I think you will have to know a lot of the basics of what we go over, as well as being able to maybe apply some knowledge. Like a question might be one or two steps away from what was presented on. Um, but that's from my understanding. Kelsey, you said you ATA'd for this course before. I'm not sure if you have any other input.
Yeah, well, when I took it, we weren't allowed to have notes or anything. So it is a lot of memorizing and just trying to make sure you understand everything. But I know the rules do change semester to semester. So I don't know. I don't think notes I'm not sure about that for this semester, but
Yeah, I'm not sure
um,
if we answered your question, Caitlin, but
okay, yeah, so just basically trying to actively recall stuff as much as you can.
yeah. And if you can, um, just however your brain works, how you know, like whether you're um, like reading or videos, just try to focus on that and for understanding how things work. Um, that's what I would try to focus on.
Okay, sounds good.
cool. Yeah, I had another office hour Monday morning, and students were also asking about kind of like what exams are going to look like. Previously, it sounds like there have been study guide materials or practice exams provided. So those will be great study tools for understanding some of what's going to be on the exam. But I think you guys are responsible for continuing to keep up with all of the different reading quizzes and especially the recitations. Those types of questions will be on the exam as well.
And also, Dr. Holcheck's office hours are very helpful, especially I don't remember what she calls them, but like the first ones after the exam, because they'll go over the most missed questions. So that can be really helpful too.
Hmm. Thanks, Kelsey. Yeah, and good question, Caitlin. That was always my worry, like at the start of every class, was not being prepared enough for the first exam. Because it's not until the first exam where you really understand like what's needed to get a good grade. So cool. Are there any other questions before I kind of go over the recitation PowerPoint? Cool. So if not, um, so the first recitation, I'm not sure if all of you have started working on this or have even looked at it. It'll just be going over the central dogma. So it'll be kind of similar to your first activity. And all of the content that's in this presentation should also be in some of the other lectures that you have already probably watched from Dr. Holacek. Um, so first things first. The learning objectives for this recitation are to understand the structure and composition of DNA and RNA, to define the central dogma of biology, and then review the history of biology and the experiments that identified DNA as genetic heritable material. So just some basics about the DNA. As you can see, DNA stands for deoxyribonucleic acid, and that's the hereditary information stored inside genes. So it's made up of nucleotide chains that are bonded together in the shape of a double helix. So as you can see, we have four bases, thymine, guanine, cytosine, and adenine, and then they base pair together and eventually form a complex structure called chromosomes within the nucleus of the cell. And that's just a good way for getting, billions of base pairs into such a small area. So then that nucleus resides within kind of the center of the cell with all the different organelles around it. And it's important to note that eukaryotes have DNA stored inside their nucleus. I'm sure if you look it up online, you'll see why prokaryotes do not have DNA in their nuclei. But yeah, there's a question on your recitation quiz about that as well. So here's just some of the basics of the actual structure of the DNA. Nucleotides have three different parts: a phosphate group, a deoxyribose sugar, and a nitrogenous base. So here's how they actually base pair. You can see that there's hydrogen bonds between the guanine and the cytosine, as well as the thymine and the adenine. And then we have the phosphate groups here labeled in green. And then you have your deoxyribose sugar attached to different nucleobases. And the bonds, the chemical bonds that link one base to another through a phosphate group, are phosphodiester bonds. And it's a type of covalent linkage. So these bonds, the phosphodiester bonds, are a bit different than the actual hydrogen bonds. So just going forward, there's also a rule that's kind of the basis for how the structure of DNA was found, and it's Chargaff's rule. And it says that adenine always pairs to thymine, while the guanine always pairs to the cytosine. And adenine and guanine are what are known as purines, while thymine and cytosine are pyrimidines. So what's important here also is that adenine and thymine have two hydrogen bonds, while guanine and cytosine have three. So that's kind of one of the main distinctions that allows for the specific bonding between these two different base pairs. And these GC interactions are also important because, as you can see, they have one extra hydrogen bond, which makes the DNA more stable. So if you're looking at a specific segment of DNA, something that has more GC interactions might have more stability than more AT interactions. Another important thing I want to point out is that this adenine, since adenine always pairs with thymine, you're always going to have the same amount of adenine and thymine in double-stranded DNA. Same thing with guanine and cytosine. So that'll also kind of relate to one of the questions you have on the quiz. And then we also want to talk about DNA versus RNA. So they're very similar, except probably three main differences I could think of off the top of my head right now. So the first is that DNA is double-stranded, while RNA is single-stranded. And then the second is that DNA is made of deoxyribose, while the sugar that's in the backbone of RNA is just ribose. So the only difference here is, as you can guess with the deoxy part of the name, you don't have an oxygen on this carbon coming off of this carbon. So because of that, that's why it's deoxyribonucleic acid. And then third of all, what you're going to notice is that thymine is present in DNA, but uracil is actually only present in RNA. So whenever you see a T in DNA, it's actually going to be a U in RNA when it's transcribed off of it. And then, of course, C, G, and A are all conserved between DNA and RNA. So here's just the central dogma as a whole. You were introduced to it, like I said, in the activity that was due yesterday. And we see that DNA is transcribed into mRNA or messenger RNA. And then that is translated into a protein that's made up of all these amino acids. So just looking at transcription overall, you see that we have the double-stranded DNA here. with both of these blue lines representing each strand with all of the different bases stemming off of it. So whenever DNA has to be transcribed, it'll open up like a bubble. And there's a bunch of different proteins that do that, that I think you might be learning about later in the course. Then off of this DNA, there's an enzyme that goes along and actually attaches different RNA, like nucleic acids, to form the actual RNA that will then be used for translating it into a protein. So yeah, over here you see that DNA is unwound, and then RNA polymerase splits the two strands and reads the DNA nucleotides. Then that synthesizes messenger RNA or mRNA. So the new strand is complementary to the template strand. So here you see the template strand is here. Complementary just means that it can base pair to one another. So for example, where my cursor is, if it's a cytosine on the template strand, you know it's going to be a guanine on the RNA strand at this position. And then it's the same as the coding strand, which is up here, which is also complementary to the template strand, but uracil is there in place of thymine. So then there's another depiction of it as well. So then with translation, this is the part that I think is really cool also, is that you have the mRNA, but then it exits the nucleus and goes into the cytoplasm where it interacts with the ribosome. So here you have the two subunits of the ribosome, kind of like those two purplish colors. And then tRNA, which stands for transfer RNA, is this cool RNA molecule that has like a tertiary structure, so it kind of folds onto itself. And each tRNA is specific for different amino acids. So then the tRNA also has an anticodon that recognizes the codon, which are the three bases right next to each other or adjacent to each other. And that leads to the addition of a specific amino acid to a growing polypeptide chain. So as you can see on the bottom right here are examples of codons and the amino acid that it encodes for or is associated with. So here is the amino acid table that was also given in activity 1. I also had a question about this. Oops, sorry. I also had a question about this on Monday. You will be provided with an amino acid table for exam, so you don't have to memorize this whatsoever. So there are 20 amino acids encoded by 61 codons. And you could see that, there might be a couple amino acids, like maybe tryptophan that I could see right off the bat, and then methionine, of course, since that's the start codon, but they are only encoded by one specific codon. Meanwhile, pretty much every other amino acid, for example, proline, leucine, all of these are encoded by multiple amino acids. So not all of the amino acids are encoded by one codon only. There can be multiple codons that encode for the same one. So yeah, like we talked about, the amino acid can be identified using this table and the 3 nucleotide mRNA codon. The ribosome is instructed when to start and to stop translation using specific codons. As you can see, there are 3 different stop codons and only 1 start codon. So these stop codons do not encode also for an amino acid. It's just a signal to actually stop. So there's no new amino acid being added to the growing polypeptide chain. So then we just want to talk about also a brief history of the experiments that lead to the discovery of DNA as the heritable material. I'm not sure what the exam questions look like this year. I don't think we have access to the exams as TAs also, but I just TA'd for Bio 340 course this past spring, and they did have a good amount of questions on the history of finding DNA as heritable material. So I would just familiarize yourself with how and why they did these experiments, which I'll cover in these next few slides. So first was the Griffith experiment, which first identified that hereditary information can be transferred from the cell or from one cell to another horizontally. I think that's supposed to say one. So he also saw that this transfer is done using a transformation factor. The way this experiment was set up was that you had a rough strain, which was non-virulent, which means that if it was injected into a mouse, it would live. So this was just a type of bacteria that was not going to actually cause a disease. So up here is a different strain of a bacteria called the smooth strain, and it is virulent. God, that's a tough word to say. So when it gets injected into the mouse dies. It looks like it's sleeping here, but that's like a nice way of putting it. Um, so then we have the heat-killed smooth strain here. So we pretty much get rid of the virulence of the smooth strain, and it makes sense that when that mouse is injected, the mouse lives. However, what Griffith and other, researchers thought was cool or interesting really was that when the rough strain, which doesn't kill the mouse, was combined with the heat-killed smooth strain, which shouldn't kill the mouse either, the mouse did die. So that indicates that somehow this rough strain was becoming virulent. And they hypothesized that something from the heat-killed smooth strain um, actually went into the rough strain and made it virulent. So they didn't know what it was, like what was actually from the smooth strain that made these bacteria deadly. So that leads to the next experiment, which was the McCarty, MacLeod, Avery experiments. And they set out to identify what this transformation factor was, and they hypothesized it was either DNA or RNA. So they attempted multiple transformations with proteins removed using proteinase. If you ever see ASE, that usually means it's like something that degrades, the word that comes before. So proteinase is a solution that destroys all those proteins. RNase is something that destroys all RNA, and DNase destroys all DNA. So they removed proteins, RNA, and DNA separately, and they noticed that the transformation from R to S only, like these are the rough cells and the smooth cells again, I believe, they only failed when DNA was absent. So this confirmed that DNA must be the transforming factor and not RNA and protein. Because when you get rid of protein and RNA, you still see transformation occurring, indicating that they're not actually the reason transformation happens. So, in science, people will have a convincing argument like these three scientists did. But for some reason, people need, validation of everything because obviously you can't see what's actually happening with your eyes. Um, this is happening on like almost a quantum level. So you need to do some sort of validation experiment. So Hershey and Chase did another experiment that validated that DNA was actually the heritable material and not protein. So they use viruses, these bacteriophages here and there, that infect the bacteria, and they labeled the protein coat of the bacteriophage with a radioactive sulfur. So this is easy to see under different lenses, like certain wavelengths will make it glow pretty much, or it'll make it radioactive, and then you can measure that as well. Or they also, in another sample, um, labeled the phosphorus in the DNA. So as you can see, they needed to label some sort of element in the protein that's not present in the DNA. So they use sulfur because sulfur is part of some amino acids. So, anyways, the outside proteins were labeled, and then the DNA were labeled. So then they allowed for infection, and then they did a blending part that actually shook the bacteriophages off of the cells, and then they were able to centrifuge this, blended liquid, and all the bacteria should, at this point, um, should have gone into the pellet. And they were able to show that after blending, um, they showed that the DNA had successfully entered the bacteria cell, and it must be the heritable material. Um, yeah, so when the bacteria pelleted, you can see that all of the proteins of the virus were still outside the bacteria, so it must not have been the transforming factor. But when the pellet was radioactive, when the DNA was labeled, you could see that since the DNA went from the bacteriophage to the bacteria, that's another line of evidence indicating that DNA is actually the transforming factor. So that's pretty much all of the slideshow. Um, if you guys have any questions about anything I just talked about, feel free to throw them at me.
I'm hopping in a little late on this, but I was wondering, do we have access to the slideshow here? Because it's slightly different than the lecture that I just took notes on.
Really? Okay, so the slideshow itself, this was just the I don't think you actually have uh access to this. This is just from a drive that the TAs have. But what
Okay.
exactly was different, if I wouldn't mind?
Oh no, it's just like the diagrams and some of the way it was explained was just slightly different, but it's the same thing, like the radioactive sulfur, the radioactive phosphorus, what the outcomes were and everything like that. It was just kind of like structured slightly different.
Gotcha. Okay. Yeah, unfortunately I didn't check if there were any more modifications made to the PowerPoint. As you can see, as I was presenting, there were a couple typos, or maybe you didn't see these early on, but there were a couple typos and um yeah, definitely. I mean, the TA who prepared this, Aaron, he definitely has like a different background to me as
Hmm.
well. So that's probably another reason why we just explained it differently.
And then because I haven't gotten to the
Okay.
recitation yet,
Yeah.
but when we do these recitations, are we gonna have like, is it just a video of something being explained, or is it like a video and something like a PowerPoint that goes along with it?
So it is just a video. Would you like the PowerPoint though? Like, do you think that would be helpful?
Yeah, I think so because I think if we had a PowerPoint too, that would like it because sometimes we might be in setting because this is a remote class. We might be in settings where we can't watch a video or we want to have like a bet like we're trying to get down what you're trying to say and we're trying to visualize like okay what
Yeah.
like graphics always usually help and stuff like that. So that'd be awesome.
Okay, that sounds good. I'll yeah, previous classes I TA'd for, we just, uploaded our slides as a PDF.
Hmm.
Um, and so I'll reach out to Dr. Holacek, and also this TA to see if we could add, a link to a PDF because I did also appreciate having slides available to look over that I could download myself. So,
Right, so this is
oh,
below the video if I remember
below the
correct.
video.
Oh, cool.
Okay, so maybe it is already there. Cool. But yeah, definitely like writing on the slides were a good idea. Like if you download them. Cool. Thank you, Lily.
Did you want to stick to questions just related to the recitation, or did you want to go over any other questions that we were kind of curious about?
So yeah, I mean, yeah, my hope was just to go over this, maybe answer some questions. I do want to make sure though that everyone feels comfortable with these experiments. I mean, it's gonna like maybe you guys have learned about these before, but these are like kind of like the hallmark experience or experiments for, how DNA was found to be the heritable material. Um,
Hmm.
and I would anticipate there being some exam questions about these as well.
Hmm.
But I don't know. When I learned this, the experiments were the hardest for me to like fully understand. Like I understood what they did, but the why was a little weird. So
Hmm.
as long as everyone feels good about that, I mean, yeah, feel free to ask questions about other assignments. From my understanding, activity two is due late, like soon as well, right?
That's a good question. I had a spreadsheet of activity two isn't due until Friday, so I guess relatively soon.
Okay, that sounds good. Cool. Okay, I'll stop sharing my screen for the PowerPoint though. And then yeah, feel free to ask any questions you have, Randy.
Yeah, I had a question. It's kind of a broader question.
Hmm.
So, I was looking at the extra credit, like the bonus project and everything like that.
Yes. Okay.
And I made sure to note some of the topics that I thought were of interest, but I also noticed that there were people kind of like signing up for the project already and already had like complete groups.
Okay.
Um, and I was curious if was this like were people meeting and discussing this on like Discord, or was it like were people kind of like um, like independently emailing each other? Like how were they?
Actually, yeah, like actually forming the groups.
Yeah, yeah.
That I'm actually not really familiar with. I'm gonna look at the bonus. So this is the what is research panel presentation summary part, and then you also like signed up for to actually be part of a group, right?
Yeah, like it's to yeah.
Gotcha. Kelsey, if you don't mind me asking you as well. So Kelsey's our ATA on the
Mhm.
call right now, and she has worked with this Bio 340 course also. Do you know
Mhm.
how people normally make their teams?
Yeah, when I was in the class, there was a spreadsheet. Let me actually, could you give me a minute to just go look at the page to make sure I'm explaining it
Oh
correctly?
yeah, of
Oh yeah.
course.
Yeah.
Yeah, there is a spreadsheet that's where people's like names and stuff are on them. I was just curious because some of those spots seem to be filling pretty quick, and I was like, I don't want to miss out on.
Yeah.
For what I did is I would look at the topics and then pick what I was interested in, and
Mhm.
then I found the topic I wanted to do. So I guess it's like first come, first serve. And then on the Discord server, there's people being like, oh, there's 2 spots left in blah 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 group. And
Mhm.
so that would get people's attention to say, hey, you're gonna have a full group and B, come join us. So it's
Mhm.
kind of both Discord and just putting your name out there.
Yeah,
Mhm.
if you see one that has open spots that you're interested in, you can just put your name in there.
Mhm.
Yeah, you don't have to ask permission from whoever's already in the group.
Oh yeah, I was just curious, like how
Yeah.
people were forming the groups, if they were
Oh, like
like,
how to communicate and stuff.
yeah, yeah, how what
Yeah,
like what's really the best way?
yeah, I think just the teams talk about whichever's best for who's involved.
Mhm.
When I did it, we texted.
Mm. Well, because I'm wondering if some of these people already know each other that like signed up with each other for some of the topics, and that's how they formed an initial group, or like they hit up people on Discord and were like, hey, if you want to sign up for this topic, you know what I mean? Like before they got in a group together. Does that make sense?
Yeah.
I do know that people are using Discord to communicate, and I think that's how they're finding each other. But otherwise, you just go in there and put your name down if that's what
Okay,
you're interested in.
gotcha.
Yeah, I feel that, Randy. I totally would have like felt like an outsider if I wasn't sure who to like actually sign up with. So
Yeah, because especially some of the topics I'm interested in, I'm like, nobody signed up for this yet. And I'm like, I don't want to be the,
yeah.
only because I think we have to have a group of at least three. So it's like, hmm. And I know I think we have to decide what the deadline is. Like it's either Saturday, Sunday, or Monday. I can't remember what the deadline is to sign up for this.
Okay. That sounds good. Yeah, so does anyone else have any questions about the course in general? I talked about activity two a little bit on Monday with someone who was asking, so I'm free to go over other stuff, even if I haven't like looked at it yet, but um yeah, just let me know.
No, I don't think it was activity 2, but I think it was the recitation quiz that's due later today, right?
Yes. Or
Okay.
yeah, it's due. I think it might actually be due tomorrow.
Yeah, I thought it says like because I have the an outline here. It's like recitation number one, central dogma. Due yeah, Thursday.
Yeah, so depending on what time it is or wherever you're at, yeah, it'll be yeah, Thursday, May 22nd at midnight.
Okay, sounds good. I'll probably work on that.
That sounds good. Yeah, I think it was I forget how many questions it is now. It's a decent amount of questions, and it is just really over the PowerPoint slides. I know when the TAs record videos on the PowerPoints, they try to, drop some hints in there on what answers might be for certain questions.
Hmm.
But yeah. Yeah, and then if you have any questions about the problems themselves, if something's worded weirdly, just feel free to reach out to us too.
Okay, well, thank you so much.
Yeah, of course. Is anyone taking any other courses this summer while taking genetics? Because from my, perspective, this is a pretty heavy workload for one course. So I was just curious.
Unfortunately, I am.
Wow, how many other courses?
Just two for session A, including genetics, and then two for B.
Wow, okay. That's a busy summer.
Well, I just finished taking 6 classes for spring, and including chemistry 1 and 2, so this is kind of
Yeah,
not as bad.
yeah, that's fair, but oh geez, 6 classes that's a lot.
Yeah, were you doing those A and B's like for last semester, or were you doing like a full semester?
I did 3 for session A, chemistry and 2 other electives, and then session B, Chem 2 and 2 other electives.
Oh man, wow. Yeah, the most I ever did for at ASU was in like A and B split was like 2 and 1 at like in like semester A and then like 3 in semester B. So I couldn't imagine like, oh my gosh. Yeah. Are you going to do organic chemistry, I'm guessing, when the fall hits?
No, I'm biological science major, but my concentration does not require OChem or biochem.
Lucky.
So for hours a day, I know with the way like credit hours are calculated, in terms of how many hours you'll be spending on a course, I think on the syllabus it says like 30 hours a week. Um, well, is what's expected. I mean, obviously, every day you're gonna have different availability for how many hours you can actually put into the course. But I would recommend, even if you're super busy one day, to try to set aside at least 45 minutes, at the minimum to stay up with what's in this course because the 6 weeks are going to go by super fast. And learning some of the complex material that's going to be thrown at you in this course will require you to kind of look at it, for sometimes two, three, but even like 5 or 6 times, and kind of like teach yourself over and over again in order to be fully prepared for exams. So some days I'm sure you'll spend like, you know, 4, 5 hours on this stuff sometimes, but other days it could be lighter, if that makes sense.
Yeah, thank you. I was just really confused because I work full time and some, like I work like 45 to 50 hour work weeks. So it's just like I'm like stressed out because I have another course for the summer as well.
Hmm.
So I'm like trying to like balance out my day with like I think my advisor recommended like side blocks or something like that sort. So I'm kind
Yeah.
of like trying to routine myself for the day, and it's kind of hard because I am the main night supervisor.
Ooh,
So
yeah.
like today I go in at 1:30 to 10 o'clock and I'm here already at work because I just wanted to get studying time in before 1 o'clock. But yeah, thank you so much for answering my question. I appreciate it.
Yeah, yeah, definitely. I mean, in general, so the lecture videos, um, that'll just be a lot of time, listening to all of the information that you're supposed to learn. But that's a little, I wouldn't call it more mindless than actually sitting down and working on a actual like activity or the recitation quizzes or whatever else there is. But yeah, those time blocks, for me, I can do an hour, maybe an hour 15 of actual like focused work before I start fizzling out. So however you can spread those out throughout your day, that'll definitely be good. Yeah, especially when you have a work schedule like that. But the lab I work in, the manager took this course online and she just said that she just didn't have weekends because, she's working, 40-hour weeks. And then on the weekends, she would have to actually kind of like bust out all of this work, if that makes sense. So, yeah.
I was gonna say that from so far from what it seems like we're what technically like the third day into this course, um, that the way it's structured, it seems kind of like it's broken up into you have readings, then there's lectures, and then basically quiz for like a chapter, and then or a module like there's 1.1, 1.2.
Mhm.
And it seems like when we get to the quizzes, the questions are uh, kind of twisted versions of like what's in the PowerPoint slides and the videos. Some are like historical questions. Some are like, specifically about the process of genetics itself.
Right. Mhm.
And when it comes to like studying, um, I think just having like practice examples. So there's probably like pre-made flashcards from people who've already been in the course, like using Quizlet, Anki, things like that. So you don't have to spend time making the flashcards. And then if some people are already creating flashcards, maybe they can make it like a group project where everybody like makes a couple flashcards, and then um, then they share it with everybody, and it's like everybody's kind of studying the same way, and there's a couple ideas out there.
Yeah. Yeah, ultimately, yeah, finding whatever works for you will be good. Is this everyone's like first upper level bio course as well, or have you guys taken other courses that are like 300 level?
I think this is my first upper level. I was trying to think there for a second. I
Gotcha.
have taken like other upper level courses, but I think for biology specifically, I think this is the first.
Gotcha. Yeah, so I didn't do my undergrad at ASU, so I'm not really sure what the distinctions were, but I remember the first like upper level bio course I took, it was a big like eye opener on like how it will require more work just to get used to um, keeping up with the type of material
Mm.
that you're doing. Yeah.
Another quick question I had is that do we have assigned TAs or can we just email any of you guys?
Oh, you could just email any of us.
Okay, cool.
Um, yeah, yeah. I mean, yeah, when it comes to anything when it comes to like grading, the TAs might be able to help depending on what assignment it is, but most of those questions should probably be sent to Dr. Holacek in terms of like trying to submit late work or anything like that. But yeah, feel free to reach out to any of us.
Can you do like a small review of 1.5? Like the tree and ancestry stuff?
Definitely. So let me open up section 1.5. Are there any specific the videos or readings that you were looking at?
Not really. I did not do the best on the quiz.
Okay.
Um, because a lot of the questions felt easy, but then the answers that were given did not match what I thought they would.
Definitely. So you're talking about like the tracing evolutionary relationships, like that type of stuff.
yeah, like the clades and all of that stuff.
That sounds good. Have you started working on Activity 2 by any chance?
Yeah, I finished that one.
Okay. Um, how'd you feel that went?
I thought okay when it comes to filling out the tables and the charts and stuff like that. I think
Okay.
I was more confused on like um the questions from the quiz
Hmm.
or like the name the definitions and stuff like that.
Okay, that sounds good. Yeah, let me open up the quiz too, just to remind myself. Sorry, I haven't actually looked at any of the
Oh, you're
concepts.
fine.
Yeah, but all of this side of genetics, I will say, is not my like strong suit. I definitely like the molecular side more. So
Yeah.
yeah. But this is also an important part of genetics. I mean, it's like the other half, so I see. So clades, here, let me share like one of the readings. Okay. So can you see that screen?
Yeah.
Okay. So kind of like, I mean, just like it says in the reading, the most commonly used approach to phylogenetic tree construction is the cladistic approach or cladistic. I don't know if I'm saying that right. Um, because, these phylogenetic trees get super complex super fast depending on how many organisms you're looking at. So when you define a clade, you're looking at a specific like characteristic that all of the animals share. So,
Okay.
um, okay, looking at this stuff, this is like, yeah, bringing flashbacks to when I learned this. Yeah, you have like your most recent common ancestor, but there might be a difference between, well, there's obviously a difference between these four organisms and this one. Maybe this one's a prokaryote that doesn't have a nucleus, and then all of these do. But then within this, there might be separate clades that distinguish organisms between one another as well. So ultimately, just knowing that a clade is a group of organisms that seem to evolve, from the same ancestor because they have a distinguishing feature that other organisms that they're being compared to don't.
Okay.
Does that make any sense? I don't know if that answered your question.
Yeah, because then you have the analogous traits and then the other trait and
Uh-huh.
trying to figure out the difference between those and which ones apply.
Definitely, definitely. Yeah, so I don't work with, evolutionary relationships too much, but another one of the TAs, I think his name's Wang Sheng. I don't know if I'm saying his name right. I don't have it right here, but he does a lot of bioinformatics works, and that deals with a lot of grouping of organisms together to look at shared genes and expression of them. So I would definitely reach out to him too, or maybe attend one of his office hours.
And he
And
does have
Okay.
office hours today,
yeah, yeah.
so
So
that's kind of perfect.
yeah, yeah. So I'm not sure. If I was a betting man, he would know more about this than me, or be able to put it in a different way, especially if he works with bioinformatics or something like that. So,
Okay. Thank you.
yeah, of course. Does anyone have anything fun planned for this weekend? It is a holiday weekend, right? I think. Well, I know it's only Wednesday, but I'm already looking forward to it. So, Kelsey, I'm not sure if you're still there, but um, for these office hours, if people usually, are done with questions, do we just hang out here kind of in silence?
You can if you want, but since it's so close to 10, you can just, I don't know. Last semester, I was with someone who, if no one showed up in the first 15 minutes, she would just close it.
Like,
So
so they would still
you can
record.
do whatever you want.
Okay, that sounds good. I just wasn't sure since, we record these office hours. It
Yeah.
should, just stay on for the full hour if that's a requirement. But that is true. Like if I started at 9 o'clock and at 9:20 even, there was nobody.
Yeah.
Okay, that sounds good. I was just curious what the expectations were.
Yeah. I'm not sure if there is a rule, but that's what was going on last semester. So
Okay. I figure if it's been done before, then it'll be all right. Yeah. I was just yeah, my first office hours on Monday, I had like 9 people show up and like they stayed for the entire time just because we were talking about random things. So
yeah.
okay. Cool though. Randy, do you have any questions? I'm not sure if you've started working on the recitation or anything like that, but if Randy's still around, I don't know. Yeah. You know. Okay. So if there's no more questions, I think I'll end the recording now.